So at the end of the 1970s, after 20 years of classic cellular immunology, we knew what the cells were, the major cells that were comprising the immune system, and those were lymphocytes and macrophages. And the lymphocytes at that time were categorized into two different varieties: the B cells that ultimately matured to become antibody-forming cells, and then the T cells, which matured in the thymus and ultimately became what we call the cellular arm of the immune system. And we knew that macrophages were somehow involved too, because if we removed them, then nothing would happen in our tests that we did in culture. So at the same time, we also knew that we had to have histocompatibility between the lymphocytes and the macrophages for anything to happen, for antibody formation, for lymphocyte proliferation, and all these different kinds of things. But we didn't know why that was. And so as 1980 neared, it became clear that what we needed to do was the same thing that the immunologists had done in the first half of the 20th century when they had discovered antibody activities, and they wanted to know, okay, so what's behind that? I mean, what are the molecules involved? And so at that time, it was clear that that's what we needed to do. We needed to reduce the complexity of populations of lymphocytes that we put into our test tubes and our culture flasks. We needed to get down to the individual cells and the individual molecules. And this was also, I had entered the field almost 10 years prior to this. But this was setting the stage for the whole thing. And so at that time, we had discovered, meaning the field in a general sense, had discovered that if you cultured lymphocytes for a while, particularly T cells, activated them so that they would proliferate, and then took the culture media from these cells and placed it on new cells, that things would happen. And this basically was called lymphocyte conditioned medium. It was conditioned by the cells that were growing in the culture flask. And so exactly what was causing all these things to be happening, there were three different assays that had evolved by then. One was the proliferation of the lymphocytes, the multiplication of the cells. So we had proliferation assays. And then another assay was for antibody formation by the cells. And we had that assay. And then we had one more assay that was concerned with showing that the cells could kill other cells if they recognized them as foreign. So we had proliferation, antibody formation, and lymphocyte-mediated cytotoxicity or killer assays. And over the course of the mid to late 70s, more and more reports began appearing that the people had been recognizing, with using one of these three different assays, different activities. And so they had different names. Each investigator came out with a different name. There must have been 20 or 30 different names published between 1975 or 1972 or 3 and 1978 or 9. And nobody knew what these things were all about. And so about in the mid 70s, people started wondering whether or not you could keep these lymphocytes proliferating in the test tube longer than normal. And then actually, the first report was out in 1974 by a friend of mine working in Lausanne, Switzerland, Rob McDonald, who showed that if he put two different lymphocytes together from two different mice strains or two different humans, that they would activate each other and they would proliferate. But they'd only proliferate for a few days. But if you then replenished the culture fluid and added more stimulating cells, then you could get them to grow longer. And he was able to get them to grow for several months. And then there was another report out of Sweden that did exactly the same thing. And then another report saying that they could grow them even longer, doing these repetitive, what they call mixed lymphocyte cultures. And so, but it wasn't until 1976 when Doris Morgan, who was working in Bob Gallo's group at the National Cancer Institute down in Bethesda, showed that if she took culture fluid from activated lymphocytes, and if she cultured and put new cells in with these activated culture fluids, that she could get cells to grow for several months without adding more stimulator cells, just adding the culture medium, replenishing that conditioned medium. And that was basically a breakthrough. And initially, they got into contact with me. My friend Frank Rossetti, who was part of that team down there, called me up and they said, Kendall, it seems like we can grow these lymphocytes for a long time with this conditioned medium, but we don't know what the cells are that are growing. And we think they're T cells because of different assays that they've done. And they knew at the time that I had an assay, because I had spent some time down in their lab. I had an assay for interferon, which is the activity that inhibits viral replication. So they wondered whether or not their cells could make interferon. And so they said, well, what we want to do is send you some supernatants and from our cultures, and then you can test them in your assay, and this will be a double-blind experiment. We won't tell you what they are beforehand, and you just tell us what the results are. And so I said, sure. And so we went ahead and did that. And the very first experiment was very clear-cut. Only the cells that had been activated properly in their hands would have had the interferon activity in our hands. There are several problems with this, because the initial cells that they got to grow long-term were derived from bone marrow samples, because what they were trying to do is that they were trying to grow leukemia cells in culture. They didn't care anything about these lymphocytes. Who cares, And so they were taking bone marrow samples from patients that had leukemia, and they were putting them into culture with a whole bunch of things to try to get the leukemia cells to grow. The problem was the cells they looked a little bit like leukemia cells, but they weren't quite sure. So that when we got our results that we collaborated on together, it was just more confirmation, along with their other tests, that these things were really T cells, the thymic-derived lymphocytes. And at the time, we had, in my lab up at Dartmouth, we had generated a system so that we could take leukemia cells. I was interested in trying to generate cytotoxic T cells that would kill leukemia cells. And I could do that. We could do that by several machinations that we went through, but we could only grow them for like 3 or 4 days. And so I said, well, Frank was on the other end of the phone, and I said, Frank, it's interesting that you can do that. I said, we'd like to see whether or not we can use that stuff to grow our cytotoxic T cells long term, and to see whether or not they would maintain their cytotoxicity, and so and that would allow us then to grow up large numbers of these crazy T cells. So we started a collaboration to do this, and these people were working in the human cells and leukemia cells and so forth, but our system was all geared for the mouse. And so we switched everything to the mouse, and we started to do some experiments along with them. And then I got a call after about a month or two. We hadn't really done very much at the time. Got a call back from Frank, and he said, Gallo wants me to discontinue any collaboration with you. And I said, really? I said, why is that? And he said, because he wants us to find the leukemia growth factor. He doesn't want to work on these T cells at all. And he's going to give the whole project to another immunology lab down here in Bethesda. And he's forbidden us to work on the T cell thing anymore. So I said, well, whatever. And you go your way, we'll go our way. And I guess the saying is the rest is history, because we took our mouse T cells that we'd activated, that we'd immunized mice with leukemia cells. And then after a while, repetitively, and then after a while, we'd take the spleen cells from these mice and mix them together with the same leukemia cells that we'd immunized with. We'd do that in test tube, in what we call in vitro. And we'd grow them for a few days. And then we would do a reactivation, a secondary mixed tumor lymphocyte culture is what we called it. And we'd wait a few days, and then we put those cells in the conditioned medium and wanted to see if they would grow. And I didn't think it was really going to work, because the dogma at the time is that the only really thing that can make T cells proliferate is the antigen, which in this case would have been a tumor antigen. And whatever the conditioned medium, whatever silly factors were in this conditioned medium were looked upon by the immunologists as just being whatever. Sort of that they called it lymphodrek. And so, so we started the experiment, and the very first experiments just worked beautifully. And we were able to grow these cells long term, and ultimately we grew them for months. And we would then test them for their cytotoxic capacity over time. And they maintained their cytotoxic capacity. And then we had an interesting visit by another investigator from Bethesda down at the NCI. And I invited him to come up to Dartmouth to talk to us, because he was the investigator that Gallo said, you take this and run with it. I'm not going to have my lab work on it anymore. And so I was sort of curious as to what this guy was doing, since at that time in the world, I think we were the only two labs in the world that really knew about this whole phenomenon and what was going on. So his name was Ronald Herbermann, and he came up to Dartmouth and gave a seminar, and then we went to lunch at the Hanover and we were talking about things, and they were having trouble repeating the Gallo, the Doris Morgan, Frank Rossetti experiments. And so I was sort of pleased with that, because I knew we could repeat it, but we did it in the mouse. We didn't. He was trying to do it in the human, like Gallo's lab done. So we were talking away at lunch, and he happened to ask us. He said, "Well, have you cloned those cells?" And what a clone is, if you look in the dictionary, a clone is the asexual progeny of a single cell. And that's important in immunology for reasons I'll get into, but we hadn't done that. And so we told him we hadn't done that. He left. He went back to Bethesda. And we started in a whole set of experiments. And one of the postdocs in the lab, Paul Baker, took this project on. And there were two basic fundamental techniques to clone cells. And he tried both of them. And one of them worked really well, and it was the simplest. And what you did was you just diluted out a culture of cells to the point where you could mathematically figure out that if you then place this very dilute culture medium containing not very many cells into culture plates, that you could show that there would be only one cell per well. And so we did that, and it worked beautifully. And we reported that, we sent a paper off to Nature at the end of 1978, and we got it back rejected. And I did the same thing that I'd done in the last time around. I said, well, I wrote them a letter and told them why they had screwed up and they should really review this paper. But in the meantime, I decided to just send it to another journal, which I did right away. And so it was published in January of 1979, and it was really it was a groundbreaking paper because of the fact that the problem in immunology had been the heterogeneity of the cell populations. You have millions of cells in your culture flask. And most immunologists wanted to know about what one cell did. They didn't want to know about the whole population. I mean, the whole population was interesting, but everybody had already done that. So to be able to have a clone of cells that they could study and do, biochemistry on it and molecular biology on it and so forth, this was huge. And so we published that paper in January of 1979, and then there was just an explosion of people that wanted to make their own T cell clones to their own favorite that would specifically react and kill their own specific antigen. And so that was the part of the breakthrough for me into the immunological community, because they started asking me to meetings to talk about this whole technique and so forth. And I knew at the time that we needed to find out what this growth factor, whatever the stuff was in the conditioned medium was that was allowing us to grow our cells. Because we got ourselves into a situation whereby we were trying to make our own conditioned medium all the time in order to get ourselves to keep our cells growing. And it was, it was sort of like we were really on the edge just about all the time. So what we needed to do was to make an assay for this growth factor so that we could purify it for this, whatever the thing was in the activity in the conditioned medium. So the good news was, the fortuitous thing, when Pasteur once said that chance favors the prepared mind. And that's a famous quote from Louis Pasteur, and it just means that if you've done your homework, then you got a much better chance of striking rich than if you haven't done your homework. And so I had done my homework, because back in the early 70s when I was in France working with Georges Maté, I was working on another growth factor, but a growth factor for red blood cells. It's called Erythropoietin, or EPO for short. And I had been collaborating with another American over there in Paris who was on sabbatical from the University of Connecticut, and his name was Torgny Fredrickson. And Torg, for short, was interested in erythroleukemia, if you can believe that. And it's sort of a misnomer, because leukemia means cancer of the white blood cells, but erythro means it's cancer of the red blood cells. And it turns out that red blood cells are regulated by a hormone that's called erythropoietin. It's made by the kidney. If the kidney doesn't get enough oxygen, it starts to put out more erythropoietin, and it makes more red blood cells. And so, because the red blood cells are the ones that carry the oxygen. So we devised a method to assay for this erythropoietin that was based on assays that I had learned a few years earlier when I had been at Dartmouth working on interferon, another of these weird activities in the culture media. And in order to do that erythropoietin assay, I took what I had learned from the virology community in terms of what you do in order to find out a quantity of anything. You do doubling dilutions. You dilute it way out, twofold, one at a time. And then what we did was culture erythropoietin responsive cells in this fluid. We waited them overnight, and then we harvested the cultures, and there were ways for us to identify how much DNA had been synthesized in these cultures, and we could get these wonderful dose response curves. That was really important because of the fact that as you diluted out a sample, the farther you can dilute it out, obviously, it means there was more in there to start with. So I just really adapted that whole thing in order to measure this growth factor that we were trying to find in this lymphocyte conditioned medium, but we used our long-term T cell lines that we called CTLL for cytotoxic T lymphocyte lines, and we just created exactly the same kind of an assay. It only took 24 hours. We could get these dilution curves of the data, and therefore we could quantify, which was extremely important, how much factor we had in this versus another preparation like that. That allowed us to sort of standardize everything, and then to grow our cells. We knew how much. We figured out how much of this stuff we needed. Well, that was the beginning. When we finally got to that stage, and we had our monoclonal T cells, we had our assay for the factor that was keeping those T cells growing. So the next obvious thing to do was to purify whatever it was in those conditioned media, so that we could get down to a single molecule. And so it was important that we were able to do this rapidly and assay multiple samples, because what we did was we did the routine biochemical techniques at the time. We first concentrated the supernatants from like liters and liters down to milliliters, 1,000-fold concentration. And then we would purify that concentrated fluid over different separative columns. One column separated out the molecules on the basis of their molecular size, and another one on the charge that they had, and so forth. And after a year or two, actually it was 1981, we had reduced the complexity of all these molecules that were floating around to an individual molecule that we could show that had a particular characteristics, its molecular size, its molecular charge, and so forth. And so we knew, and we could show and demonstrate through other methods, that this activity that we had was really due to a single molecule. And that's sort of like when the antibody people had finally figured out what the structure, what the molecule was that had the antibody activity. Then by 1981 or so, 82, we had done the same thing with by then we named the growth factor as a T cell growth factor, or for short TCGF, because that was a good name, and we thought, because that told you what it did. It said its name. And so we were quite smug at the time, thinking how good we were. And it was right at that time when we finally got these. We had the growth factor, we had the monoclonal T cells, and we said to ourselves, well, because the big question at the time, and I think I went through this before, the big question was, what's the nature of the T cell antigen receptor? Nobody knew what it was, and everybody was, all the big famous labs were working on this whole thing. And we talked about it, and myself with the rest of the people in my lab, and we said, well, it's obvious what you do. You take those cells that are monoclonal T cells. They should only have one receptor, will recognize one antigen. And therefore you can immunize mice with these cells and get an antibody that will react only to this T cell clone. Now you can do that, then that's a eureka moment. And I thought about that for a while, and I thought about how to go about it, and so forth and so on. But at the time, I thought to myself, there's other things that I want to do right now. And I don't want to get involved in that, because it's going to take me. I think what we really need, the real money in this whole process is the nature, the molecular nature of the growth factor, and then how it causes the cells to grow. Because if you recall, I initially started all these experiments back maybe even a decade earlier, because I wanted to understand what regulates normal cell growth, so that I could ultimately understand what regulates cancer cell growth. And I felt that we couldn't understand the abnormal until we understood the normal. So this is where I was sort of laser focused on these kinds of questions. So I left. So I didn't chase the T cell receptor, but my good friend Ellis Reinhardt's down at Harvard, because I was up in Hanover, New Hampshire, he went ahead and did that. And what he'd done over the course of just a few years between like 1978, 9, 80, 81, 82, he characterized antibodies that had been derived by others to show that he could find one antibody would recognize all T cells in the human, and another antibody that would recognize like 2/3 of them, and then another antibody would recognize only 1/3 of them. And then he looked at normal immature cells in the thymus. Remember, the thymus is where T cells go to get educated. And he found out that in contrast to the cells, the mature ones out in the periphery, in the lymph nodes and in the blood and so forth and so on, they had markers for both the 2/3 of the lymphocytes as well as 1/3 of the lymphocytes. So they were what we call double positives. And the molecules were just labeled T4 on the one hand, that was for 2/3, and T8. And the other antibody that recognized all of the T cells was called T3. And he had shown by this time that the T3 molecule seemed to be involved in signaling through the antigen receptor, because he had an antibody that would block the T3 cells, the T cells from proliferating. Well, he took his T cell clones. He generated cytotoxic T cells from all of these cells, from the T4s and the T8s. And he took these cells and he immunized other mice. He selected out then antibodies from these mice that he'd immunized. And he ultimately made monoclonal antibodies that recognized the T cell receptor on his T cell clones. He then used these antibodies to show biochemically what the antibodies were reacting with. And he published that in the spring of 1983, and that was really the first demonstration of what the T cell antigen receptor was. And the other thing that I liked about his experiments is that his very first paper, in I think it was April of 1983, he was first in the business. He was correct in all of his interpretations of what was really going on. And he was complete. He had the whole complete ball of wax because of the fact that it turns out the T cell receptor is pretty complicated. It's comprised of several different chains. He had characterized all of the different chains, and he figured out what they do and what their molecular sizes were and so forth and so on. And he had competitors at the time. People were crawling all over the place trying to do what he did. And so it was pretty tense, for probably still is, because now different people are claiming that they discovered the T cell receptor, So anyway, by that time, it was in 1983, in the summer of 1983, the International Immunology Congress was held in Kyoto, Japan. And I went to the meeting, and Ellis was also there. And actually, and so we started hanging out together and going to bars and after the meeting and so forth and so on, and started talking about all these different aspects of what science was all about and so forth. And I told him, and I said, well, it's not the T cell receptor that does that does all the magical things. It just sets the stage. It just activates the cells so that then they do what T cell growth factor does. And so we devised a series of experiments to do, the two labs together in collaboration. And from my end of things, my laboratory, there was a postdoc that had just come to the lab, and her name was Doreen Cantrell, and she was from Nottingham, just gotten her PhD in England at Nottingham. And we went down to Boston and visited with Ellis and all of his lab, and we talked over these experiments. And then we got together and went back and did them, because the hypothesis was, well, the T cell antigen receptor, what it does is causes the cells to make the T cell growth factor. And it also causes the cells to place on their surface T cell growth factor receptors. We figured there had to be a receptor. And we had all the reagents that we needed at that point. And I left out one part of the story is that by that time as well, we had purified IL-2 to homogeneity, and we had radiolabeled IL-2 with radiolabeled amino acids so that we could detect where it was. And the hypothesis in that instance was that it was on the surface of cells. Only the antigen activated T cells, so the T cell receptor activated cells. And we had found an antibody that would react to the T cell growth factor receptor. So we had everything we needed, and we did a series of experiments that didn't take very long, and we published another paper saying that this is what's going on, everybody, that the T cell antigen receptor, the way it works is that it activates a molecular mechanism that then takes over and directs the cells to proliferate and to do their deal. I don't think that was ever really readily accepted by most of the immunologic community, because they didn't want these lymphodirect things to become characterized and have some substance, because they hadn't, it was a sort of the NIH or not invented here kind of idea. And so the thing was, at the same time, there were there were the T cell antigen receptor people, and then there were the lymphodirect people, and there were other lymphokines that had been active, that had been described and were out there. And one of which had been described at least almost a decade earlier, in 1972. That was called lymphocyte activating factor. And it was shown at that time to be a product of macrophages. And we knew that macrophages were necessary for all this business, and we set up assays to find out. We didn't know which cells were making the T cell growth factor. And so we set up assays to try to ascertain that. And we couldn't do that. And so we collaborated with another group of investigators also at the NIH, one fellow by the name of Joe Oppenheim, and then another fellow by the name of Larry Lockman from MD Anderson down in Texas, who were lymphocyte activating factor gurus. And so lymphocyte activating factor was abbreviated LAF or LAF. So what we in our lab, we said, well, LAF is a joke. And we said that because when we put LAF into our T cell growth factor assays, it fell flat on its face, didn't work at all. On the other hand, there was another assay that was out there for LAF that had been described earlier. When we put our T cell growth factor samples in the purified, the most purified that we had, they worked in the LAF assay, and so they worked in the LAF assay and in the T cell growth factor assay. So we devised a series of experiments to do with Joe Oppenheim and Larry, because they had purified LAF to what they thought was pretty close to homogeneity. So they sent some samples up to Dartmouth, and we performed the assays, and it was clear cut. And what happened was that the LAF factor, the LAF, activated the lymphocytes to make oodles of T cell growth factor. But LAF itself wouldn't cause the T cells to proliferate. Only the T cell growth factor would have caused them to proliferate. So we discussed it and so forth, and we said what we thought was the obvious thing to do. And the obvious thing to do in this instance was to say, well, LAF then causes T cells to make T cell growth factor. And there was a meeting, the second International Lymphokine Workshop, that happened at Lake Constance, which was sort of right between, it was in Germany, I think, or Switzerland, I can't remember, right on the border between Germany and Switzerland. in the summer, and we took all of our data and presented it at that meeting. And so that activated all the lymphodirect people. And we stayed up late at night drinking beer and discussing what we ought to do with all these data and what was really going on, and with all kinds of people. But this time there were multiple people involved in the whole thing. And ultimately we decided that we should, well, I shouldn't say we decided. Ultimately it was decided by this group of young investigators who were clamoring over each other to change the names of LAF and TCGF. And so we made up a new name that was called Interleukin. Inter meaning between, and leukin meaning white blood cells. And so because LAF seemed to be acting first in a sequence of events, LAF became interleukin 1 and TCGF became interleukin 2. Which was great, and everybody agreed on that, and so forth. I never thought we should do that, but that's what happened. It was a political. Everybody else wanted to do that, and one of the reasons that they wanted to do that was that then they didn't have to reference all of our previous papers. They could just start at that time then talking about IL-1 and IL-2 and so forth and so on. But anyway, I'm having a senior moment here. Wait a second.
Take your time. It's all good.
Well, it's fun, all these stories.
I'm enjoying it. It's putting a lot of things in context again, and especially having fresh in my mind the first chapter. Since I've been editing that conversation, and so going right into this, it's like, okay, you're getting the logical sequence of events and why things were quite so important. So
later on, after the dust cleared and interleukin 1 was the accepted and interleukin 2 was the other accepted, and there wasn't an interleukin 3 yet, I told my kids, who were like 10 and 11 at the time, that I discovered the first interleukin molecule. And they said, yes, you're dead. That's why it's IL-2, right? And so, but the problem was at the time, we had purified IL-2, so we knew what the molecular characteristics were, but we hadn't gotten enough of it so that we could then use it to do more assays and to do more things. And so we needed to have another way to ramp things up, to scale up, so that we could have lots and lots of purified molecules, so that we could do both biochemistry on the purified molecules and find out more about them and so forth. But in addition, use those purified molecules in different kinds of assays to find out exactly what this stuff does and how it did it and so forth. And also LAF at the time, IL-1, wasn't as far enough along in the purification scheme of things. So it was still an activity. We were almost there, but we weren't quite there. And it took another year or two for the IL-1 people to purify their molecule and to characterize it and so forth and so on. The experiments that were important that we did at that time were number one, we were able to really characterize the interleukin 2 receptor using our radio-labeled interleukin 2. And it turns out at that time, another interesting person entered my life anyway. It was another professor at Dartmouth who was just upstairs on the 4th floor. I was on the 3rd floor. And his name was Alan Monk, and he was, 10 or 15 or 20 years my senior. He was already established in the endocrinological field because he had first identified one of the steroid hormone receptors, the receptor for glucocorticoid hormones. And so I was interested in those things too. And I may have talked about this before, but we started collaborating with Alan. And so Alan because he was an endocrinologist, an experimental endocrinologist, he basically led us through a series of steps in order to develop an assay for the IL-2 receptor. And that's what we did. And so at that time, and we published those data right about the same time, around 1981, 82 or so. So we had the IL-2 receptor assay at this stage. Now it's changed its name from T cell growth factor to IL-2. And at that point, we then took off on another series of experiments that we will get into in the chapters to come. But to round things out in this particular segment, what happened next was another revolution in biology and immunology, and that was the ability to clone genes. So we had, and that assay had been developed, or the ability to clone genes had been developed at Stanford and UCSF in the mid-1970s, and so like 1973, 74, 75. And for several years, it was pretty esoteric. Only the real molecular biology jockeys could do this sorts of stuff. The rest of us biologists who were farming around in the soups, we really couldn't do that high-powered science. And so, but an investigator from Japan by the name of Tada Taniguchi had collaborated with another group in Japan, and they wanted to his expertise to see if they could clone the gene for Interleukin-2. And they went to him and they said, well, this is what we want to do. And his comment was, well, what's Interleukin-2? He'd never really heard of it. He knew about interferon, another one of those intermolecules. And so he had cloned one of the interferon genes, and there were several. And so he set to work to do that. And at the same time I was working on that at Dartmouth, and in collaboration with another investigator down at the NIH, who was more of a gene jockey than I was. And we were working along, and we were doing pretty well. But we got beat by Taniguchi in the end. And he published in 1983 the gene, the complementary DNA that encoded the IL-2 molecule. And then we together, and he also simultaneously found the original genome, the gene that codes for the complementary DNA. And that was really, really important, because what it allowed us to do, now we could make bacteria make as much of this IL-2 as you would want. I mean, milligrams and grams and kilograms if you wanted it. All you had to do was grow bacteria in cultures, and they were really pretty easy to grow. Not like our lymphocytes that were more difficult to grow. And so it allowed us to then move to the next step, which was to start to do experiments as to how the gene coding for Interleukin-2 was regulated, turned off, turned on. And we knew that that's what was going on from our experiments with Reinhardt's, because it wasn't until you activated the T cell receptor that then you got the cells to make Interleukin-2. And it was the same way with the IL-2 receptor. It was all being activated or initiated by the T cell antigen receptor. And so then we and others then went on to identify the what they call transcription factors, which are the molecules inside the cell that get activated by the IL-2, by the T cell receptor that turn on the IL-2 gene. And all this became very, very important downstream because of the fact that regulating the production or the activity of the IL-2 molecule was the way that you could then manipulate therapeutically the immune system. And so I will stop there, I think.